Just when you finish, we want to make sure that you don't log up too quickly, because I have to grab your local recorded file.
Okay.
Yeah.
That's what I'm projecting. That's good.
Dennis, I think it may be a blessing in disguise. Okay. Yeah. Perfect. Yeah, that's right. Yeah, not better about it at all, actually. I just yeah. So the only thing I was thinking is we should get an approach to expressing interest by having a volume. I knew what they were looking for. Yeah. Okay. You know, I had a conversation about that too. So I was suggesting that. Yeah, no, I agree. I think it's going to initiate. But I think he didn't have time. But he knew the background. Yeah. Yes. Don't get me wrong.
Does this thing have a penultimate?
I mean, anyway, these days, but yeah.
Do I need to wear a mic for this? Will it just pick
I think it may be better if you wear a mic. Audio may be better. Why would we wear a lapel mic?
Yeah. I just like to see a penultimate mic on that.
This makes sure that it's actually right.
Test.
I think we're supposed to start at 12 sharp.
Okay. Start at 12 sharp.
I'm the crap.
It's kind of like if a tree falls in the forest and no one's there to hear it. I thought that's my lecturing experience yesterday. If no one attended, yeah, did you bring fruit?
How's it going?
See? It's like the last thing I needed to do was to volunteer to give a seminar that I hadn't written. So anyway, it's all good.
My Antarctic installation. This guy, stop. I'm over it.
Ready? Possibly go wrong.
Lots of things.
How many of the shirts do you have, John?
No, he's got 15 of those,
I
Craig.
guess not. It's like, see, where, I know it's not the same shirt every day.
I have 4 of those.
Day, Wednesday. Tuesday.
I don't wear this every day. I don't wear this every day.
It's really nice. They're really nice shirts, Charlie.
They have a roof. They have.
Okay, I'll just turn that one off.
Can I get the water?
It's just how many line off. It's looking.
Really?
So I just switch that one off, maybe.
Yeah.
It's just inhale.
It's a short breath thing.
Loudest thing in the room besides the speaker is huge.
How long do I have?
I see, just maybe don't go over an hour.
I will not go over an hour. I'll try to keep it fairly high level.
We were supposed to go down last week and got delayed. We're still delayed until next week. So it's probably 2 weeks behind. It just doesn't
Yeah, well, 3 more minutes.
Yes.
Comes back on.
I was gonna leave it.
Can't switch it off.
It's all right. No, I switched it off. It just has a mind of its own.
Yeah, no, it's so screwed. It's like,
That's all right. We'll just leave it. We'll just leave it.
That's good. 3 for the sheet.
Oh, yeah, I've had that. And they also like,
Well, COVID just. What else?
for the people on Zoom, we'll start, give people another couple of minutes to come in before we start. So please sit tight.
Right now. She's also speaks projecting. She
Really frustrating. They're just coming down like a few minutes.
That's all right.
it's not managed anywhere.
It's little papers. Because it's the gene. Sequence the gene. It's just it's got attributes.
Okay, we might as well get started.
Yeah,
Everybody should know me and Conrad, but I'll give an introduction anyway. I'm the academic co-director of the Waikato, which is the Environmental Research Institute. It is my pleasure today to introduce Professor Conrad Pilditch. He's a professor within the School of Science and an academic team leader. And he is a marine scientist with a specialization in coastal benthic marine ecosystems. And he has been working a lot on both natural and anthropogenic change and what the impact of those changes are to our coastal ecosystems. So today he will give us a talk on the erosion of resilience in coastal ecosystems.
Kia ora, everybody. Thanks for the introduction, Charlie, and thank you for attending, those that are online and also those that are live here in Hamilton. Appreciate having an audience. It's certainly better than I've been doing in most of my lectures lately, so I wouldn't have taken it personally if no one had shown up. So thank you all for being here, and thank you for the opportunity to share a little bit of the research that we've been involved with, or I've been involved with over the last couple of years, it's been a good opportunity to bring some of this together and share some of the findings with you. I kind of need to acknowledge a whole lot of people. I want to do this first in case I run out of time. And this has certainly been a team effort, and it's involved a lot of people. And I just want to highlight a couple of really key people who's we've been working with for a long period of time now on this kind of stuff, and who've contributed perhaps most, although there are many more that have contributed in other ways as well. And these are Rebecca Gladstone Gallagher. She was a postdoc with us for a couple of years, formerly a PhD student here now at the University of Auckland. Johanna Garmel, who has just finished a postdoc here on this project, who's gone back to the University of Helsinki, and then Judy Hewitt and Simon Thrush at the University of Auckland, and a couple of PhD students who have contributed an awful lot to what I'm going to talk about today. Reading between the lines is mostly their work. And that's Steph Mangan, who's down at NIWA in Christchurch now, and Georgina Flowers, who's a PhD student with us currently. Okay, so with that, oh, and of course, I need to acknowledge the National Science Challenge Sustainable Seas that have funded the bulk of what I'm going to talk about today. So if we think about our coastline environments, we have some 300 plus estuaries scattered along the coastline of Aotearoa. Now, these estuaries are highly diverse environments. They range from wetlands, drowned river valleys, to the coastal embayments that are kind of characterized estuary types that we have here, and of course, all the way down into the southern Fiordland area, which are actually estuaries as well. Now, these estuaries are characterized by regions of very, very high productivity. They have, because they're close to where people live in many cases, I think 24 of our 25 cities are on the coast, and most of those are based around estuarine environments. And they're based, and people have been connected with estuaries for millennia. They have high cultural values today, recreational food harvesting sources. They're intrinsic ecosystems that support a lot of biodiversity and a lot of productivity. And of course, they're the centres of commerce for a lot of our large cities, places like Tamaki Makaurau, Auckland, Tauranga Harbour, big commercial centres all based on estuaries. And that's reflected globally, where a lot of the large industrial areas are focused on these estuaries. Now, these estuaries have an immense amount of value to people, but it's underpinned by being a very productive ecosystem, first and foremost. So the seafloor in our estuaries, which is where most of the biomass and most of the productivity occurs, contains 90% of the biomass that we find in estuaries. And this is just a photograph of one of the estuaries up north of Auckland, the Whangateo Harbour. So if we look at these estuaries, they're very productive. If we look at a dense area of shellfish beds, which you can see in this photograph here, they actually represent, what do we lost, Charlie?
Sound. That's causing a bit of echo.
Okay, too loud. It's usually a first case. So these intertidal sand flats that we can see here actually contain productive shellfish beds, mainly Ostrovenus dutchburii in this case, a small clam that feeds and lives just beneath the sediment surface. So in a typical area on this sand flat with these shellfish beds, it will contain 80,000 kilograms of shellfish biomass. So that's a lot of animal biomass. If we compare that to an average dairy farm here in New Zealand, that will support about 2,000 kilograms of cow per hectare. So there's a multiplier of 40 added on to the amount of animal biomass that these intertidal habitats can maintain and produce. And there's no added nitrogen or fertilizer to these systems. It's all relatively natural. So they're incredibly productive ecosystems. And at the heart of that ecosystem is a hidden garden. And the hidden garden is comprised of microphyta benthos, which are small, single or multicellular phytoplankton that live associated with the sediments. So this is this green tinge that you can see on the sediment surface here. It's composed primarily of diatoms, which are small single-celled phytoplankton. They're attached to the sediment surface. Here's a scanning electron micrograph of one just here. So these systems are so productive for two reasons. One, there's an abundance of light that's hitting that sediment surface, so that can sustain high production. And the second is an abundance of nutrients, primarily nitrogen. And they're situated at the sediment surface, not in the water column in many of New Zealand estuaries, because the nutrients are stored in the sediments. So there's an abundance of nitrogen being regenerated by the bacteria in those sediments that supplies lots of ammonia back up towards the surface, and it's trapped there at that layer by the microphyta benthos. Now, these diatoms are naturally high in polyunsaturated fats and lipids and proteins, which makes the conversion of that plant material into animal biomass very, very efficient. And that transfer occurs through the invertebrate community that's hanging out in the sediments. The clams, the shrimps, the crabs, the whelks, all of those animals and the polychaetes are either eating directly on that microphyta benthos or feeding on the animals that are feeding on that microphyta benthos. So it's a very efficient transfer because of that energy is nicely packaged, it's high in protein, high in lipids, up into the animal biomass, and then finally up into the charismatic megafauna that many people associate with those estuaries. So it's that food source really is why the animal biomass can be so much higher in estuaries than it is in terrestrial systems. You don't need 7 stomachs and multiple different sorts of bacteria to convert cellulose into animal biomass. So these estuaries, I'm going to come back to these food webs and these interactions between the animals that live in these sediments and the productivity of these systems, because that underpins the functionality and the services that we provide for these coastal environments. Now, estuaries are probably the most contested space that we have. They're subjected to multiple stresses, and those stresses can either originate on land or in the sea, or increasingly at global scales. So from land, the estuaries are the receiving environments of contaminants that run off our terrestrial ecosystems. That's things like nutrients, things like sediments for poor land use management, but there may be point sources associated with urban areas such as wastewater treatment plants, and also the contaminants, heavy metals that run off our roads and off our cities into these environments. There's also stresses that originate in the sea. Some of our larger estuaries have aquaculture operations, maybe adding organic matter to the sediments. Fishing, either in the estuaries or on the open coast, can impact those ecosystems as well. Occasionally, we have the odd ship run aground and deposit oil into our estuarine systems as well. Many of our larger estuaries, especially those associated with dense human populations, have undergone a lot of hydrological re-engineering, dredging for shipping channels, and all of those can alter the hydrography and put stress on the ecosystem. And of course, increasingly, we're living in a warming world and a more acidified ocean as well, which adds into that system. Now, why we're concerned about these multiple stresses is that they often interact in non-additive ways. And so this means that if we have stressor A and stressor B occurring in the same place at the same time, the effect on the ecosystem may be multiplicative and not additive. And a lot of these stresses, particularly the land runoff ones, are diffuse stresses that operate over very long temporal scales and very large spatial scales. So understanding their effects on how they affect those ecosystems is particularly challenging. What we do know is that these cumulative effects in multiple stresses can lead to tipping points. And this represents these rapid changes in state and structure, and often come as a surprise to managers and surprises to society. We often know when we've passed a tipping point and ended up in a degraded state from a relatively pristine estuary like this one here, to one that may be dominated by mudflats with lower values and services and productivity. So these cumulative stresses, the cumulative effects of these multiple stresses begins to erode resilience in these systems. And what makes that? Makes them vulnerable to these rapid state changes. So often a small effect can have devastating consequences. And these changes, these state changes are often only apparent when you're sitting back down here at the bottom of the cliff, looking back up, and then, oh, it's obvious what was happening. So there's plenty of theory around how these state changes occur, what triggers these state changes. There's a lot of computer modeling being done on these kinds of things, but getting outside and demonstrating this empirically is often a much more challenging task. We want to avoid these tipping points because when we've transgressed them, getting back to that original state is particularly difficult because of the hysteresis involved in the system. So we may be able to manage the stresses, turn off the taps, but just by doing that, because of the legacy effects of these stresses, the system may not return back to that state, even though we've managed the initial causes of it. So there's real value in kind of we're moving away from these theoretical ideas of tipping points to actually getting outside and thinking at scale about how these stresses interact and providing empirical evidence for actual effects in erosional resilience. We don't want to end up down here saying, we told you so. We want to be back and be able to provide advice and evidence that these things are real and occurring. So the project that we've been running within the Sustainable Seas has been about gathering this empirical evidence. And it's been about gathering empirical evidence at scale. So something that's generalizable across the country. So not necessarily just working at one place and at one time, but increasing the scale over which we're running these studies, these experiments. So I'm going to focus, most of what I want to talk to today about is on this tipping points experiment. So we chose two stresses to have a look at. A lot of empirical research today often involves a stressor in isolation. We look at stressor A, its impact, and then stressor B, and try and make some statements about how these things are going to interact. So we chose two stresses to work on, probably the two most difficult, but also probably the two most important, at least in a New Zealand context. So we focused on the effects of sediments and the effects of nutrients as two key stresses. So here in Aotearoa, New Zealand, we do a really good job of pumping a lot of sediment into the coastal zone. This photograph here on your left is a photograph of the Manawatu after a rainfall event in the Wellington region a number of years ago. You can see the vast amounts of erosion that have occurred during that rain event. And then several days later, the satellite imagery of the lower North Island showing the results of that rainfall event in the nearshore zone. You can see the high levels of turbidity or suspended sediments occupying that nearshore region. So that's of concern because, as we know, our hidden garden likes lots of light at the seafloor. So adding that suspended sediment into the system reduces the amount of light availability at the seafloor. That's the base of the food chain and consequences through the ecosystem for that. So we've done a really good job about upping the amount of sediments we deliver to the coastal zone. Currently, we occupy about 0.05% of the land surface area on the planet, but contribute 40 times that loading to the sediment loads into the system. So we deliver about 20 times 10 to the 9 tons per year. That's a lot of sediment. Now, some of that's natural. We have steep landscapes. We live in the westerly wind belt, so we have a lot of rain. We have highly erodible soils, especially in the eastern parts of the North Island, volcanic soils. So all of them predispose for a lot of sediment coming into the system. Catchments are short. When it rains, the sediments arrive in the coastal zone fairly quickly. But our land use choices have greatly contributed to that. So recent study looking at sedimentation rates in many New Zealand estuaries has demonstrated quite clearly an order of magnitude increase in sedimentation in estuaries since European colonisation. And that really hasn't abated. The other stressor that's been growing a lot over the last kind of several decades is the intensification of agriculture on our land. And there's many in this ERI Research Institute who can talk to that much better than I can. But this simple graph here just shows the increase in the dairy herd size or the number of cows in New Zealand since the 70s, which is around 3 million to over 6 million cows on the land today. Now, given that each cow produces about the same effluent as 20 people, that's 60 million people we have on the land effectively. And of course, there's the added nutrients that are needed to keep the grass growing to feed the cows, that milk production. So some of those nutrients eventually end up in our waterways and eventually make their way down to the coast. So these stresses are difficult to deal with at scale. They're diffuse, they come in episodically, and it's a very difficult system if we want to manage or experiment with this or do empirical research. How do we go about setting up a study that will allow us to address those things? So this was the tipping points experiment that we set up. We worked across 15 estuaries in New Zealand. So we're trying to look at before we fell off a cliff, before we're in a really degraded state. We do have some estuaries in New Zealand that have passed tipping points, are now eutrophied or filled with mud. But the estuaries we selected are all in a relatively pristine state. So we're working back at the start of that place before the system actually fell off a cliff or we entered into a degraded state. So these sites were selected along a turbidity gradient where we used our collective knowledge of the estuaries we were working in and the time we'd spent working in them to pick sites that were relatively clear estuaries that didn't have a lot of suspended sediments in them, and those that had more suspended sediment loads. The communities of animals in the sediments were relatively similar. They had two key bivalve species in there, a clam and a deposit-feeding red shell. And we went into these systems and manipulated the amount of nutrients that were in the sediments. And we did that simply by adding fertilizer at depth to simulate a higher nutrient load. So we had a gradient in turbidity, which we went in and then manipulated the amount of nutrients in the sediments and then looked at the effects that it had on the ecosystem. So we set this experiment up at all these sites, cast of thousands, dug hundreds and hundreds of holes and sand flats to add the fertilizer. And then 9 months later, we went back with our benthic chambers, which are these sort of incubation chambers we place over the sediment water interface. And we can use that to measure a lot of fluxes and exchange of solutes between the water column and the sediments. We could measure things like denitrification rates. We could measure primary production. We can measure the regeneration of the nutrients and the consumption of oxygen in these sediments, all indicators of how the system is functioning. And we also sampled the sediments for the macrofaunal diversity and how the stress of turbidity and how the nutrient additions altered that biodiversity, which could then link back into the functioning of these systems. So there's some data looking at the light levels in these harbors that we went through, and you can see, although there's a lot of variability, we can see we did achieve a gradient across these 22 sites we had in the estuaries. Some estuaries had very, very low light levels. This is in Maharangi, where others in Avon Heathcote and Tauranga had reasonably high light levels within the system. So we achieved a nice gradient of turbidity across these 22 sites, which was good because that was half of our experiment. So the next thing is, how did the nutrient stress into these systems begin to change how these systems would function? So what I've got on this next slide here is an ecological interaction network. And so what this does is it represents a map, if you like, or a summary of how we think these soft sediment ecosystems are functioning and working. That interaction network is based on about 20 years of research looking at different components of this and how it affects the system. So we have parts of the system that are environmental. This is our nutrient addition here, sediment temperature, because we're working from Northland to Southland. The amount of light attenuation, turbidities in here. These are our primary producers. There's some properties of the sediment through here. And then these components, Ostrovenus and Macamona, are our two dominant shellfish species within the system. And then we've got some measures of nutrient processing in these orange boxes, which represent ammonium flux and also denitrification. Now, the details of this really aren't important. It's just a summary of how we think things are connected and how how we think things work and how we think things work. And so what we're interested in then is if we take our measures of ecosystem function and have a look at how that interaction network is going to change under turbidity stress. So we can run some boosted regression tree analyses, which can split our sites based on turbidity into clear estuaries and into turbid estuaries, and we can have a look at how that interaction network begins to change. So if we're thinking about the interaction network, we can see on the left-hand side, we've got our clear estuaries. So this is estuaries that on average had greater than 420 micromolar per meter squared of photons hitting it, and our more turbid estuaries, which is less than 350. I want you to remember these numbers because I'm going to come back to these towards the end of the talk. So there's a clear breakpoint in those ecological interaction networks around light levels in these estuaries. So again, don't want you to get bogged down in details. I just want you to visually have a look at how those interaction networks have shifted. So in clear estuaries, you can see, there's a lot of connections. There's strong connections indicated by the solid lines. There's weaker connections within the system, indicated by the dotted lines. There's positive effects and there's negative effects. It's wired. If we compare that to the more turbid systems, you can see that interaction network is simplified considerably. There's a few direct, strong interactions, but the nutrient addition is now decoupled from that interaction network where it's connected in the other one. And so if you're thinking about network theory, when you simplify networks and you reduce the number of pathways, the number of feedbacks and alternative paths, you're reducing resilience in these systems to cope with further stress. I kind of like to think of this picture maybe as the first one pre-COVID transport networks down in New Zealand. You know, we had many ships coming from different parts of the world. If one shipping route kind of fell over, then there's another way in which to get our goods in and out of the country. Post-COVID, now post-Ukraine, we're in the middle of a war in Europe. Those networks have been dramatically reduced. We're dependent on a few key connections. If something happens to that, we're isolated. And similar in the ecosystems, if we're simplifying those networks, the ability to absorb stress, the ability to find different pathways around it are now impacted. So this provides some really nice evidence in systems that are stressed by turbidity, their ability to process those nutrients is reduced and makes them more vulnerable to change. We've also got within our system a whole lot of biodiversity. And if we think about just taking a simple core from the sand flat, it may have 30 or 40 species in it. Across an area the size of this room, we may have 120 different species of macrofauna. Now these things are, you know, they look like worms and crabs, which they are, but they carry out functions and they're really, really important to the system because these little guys that are moving through their behavior, whether they're feeding, whether they're moving sediment up or down, whether they're irrigating burrows, all affect those key microbial processes. The regeneration of nutrients that fuels the primary production and the availability of oxygen within the sediment. So these macrofauna play a really important role through their mixing and their feeding processes. And so we're interested in having a look at how these stresses that we place into the system across this large scale of New Zealand, how they're influencing the biodiversity, and could we relate that back to some measures of ecosystem function. So again, collapsing a whole lot of work down into some very, very simple graphs. We found that our turbidity, and that's again, we separated this into 3 different light levels now. We have our clear estuary over here, our moderately turbid group here, and our turbid group, which had less than 350 micromolar photons per meter squared per second out there. So this is just simply a very simple measure of diversity. It's just the number of species we had in the core. And we can see as we went from the clear to the moderately turbid and the turbid, there was a slight increase in the number of species that we would find. Okay, so we had slightly more species we found in these moderately turbid and these turbid systems. We also found that as we added nutrient stress to these systems, there was a negative effect on that biodiversity, particularly in the high levels of urea addition. We dropped the diversity down from about 22 species per core down to 16 or 17 or something. So we're knocking species out with that nutrient addition. There was no interaction effect on the species richness. And so we wanted to now know how that was going to impact on the multifunctionality of the system. So what we've got here is a kind of summary of the sort of key findings from this analysis. So we calculated a multifunctionality index. Again, the details really aren't that important, but we took several measures of ecosystem functioning. There was rates of primary production, how fast carbon was being remineralized in the system, the amount of chlorophyll or primary producer biomass in the system. So we combined that into one metric, which was to give us an idea of how well the system was functioning. And then we looked at the relationship between the number of species that we're finding and that particular index. So what you can take away in this, and that those numbers of species were being reduced down in these systems by increasing nutrient stress, which we added into the system. So what you can see very clearly in the moderately turbid and our turbid sites is that as that system became stressed, we were losing species, and as a consequence of that loss of species, we were losing functionality within that ecosystem. Okay, so this is data taken from 22 sites across the whole length of New Zealand to build this relationship. So the generality of it, I think, is pretty reasonable. And it's no surprising there. We all know that biodiversity underpins function. If we lose biodiversity, then we're going to lose functionality. The interesting thing was, in the clear estuaries, we found no relationship between the loss of biodiversity and a decrease in multifunctionality. It was resilient to biodiversity loss. So as we stress the system with turbidity, as we lose species due to nutrient stress, then we saw a loss in functionality. And the clearer systems, that loss of species, as we knock species out, didn't have an impact on that multifunctionality index, which was kind of interesting. So getting back to our species again, the thing about it is this resilience in the clearer estuaries is probably due to some level of redundancy. So when we look at species, we see multiple different species in this thing. But when we start thinking about the traits that these animals have, they share certain functional traits. For example, the worms that we may see here move through and biotubate sediment. So they share the same functional traits. So functionally, if you lose one species of worm, it could be replaced, or the role that that worm was playing in maintaining functionality may be replaced by another. So you have some measure of resilience. There's different species, but doing the same jobs. So if you have high levels of functional diversity, then maybe you can begin to lose species and not impact upon that system. So we can classify these animals not in terms of whether they're species, but we can characterize the community in terms of its functional traits. And we can characterize the community in terms of its functional traits through its contribution to ecosystem function. So these are the things that the animals are doing to maintain the functionality and the productivity, the nutrient regeneration, supporting the microphyta benthos. But we could also think about traits that provide response diversity, which is how it deals with stress. Okay, so these are traits that are related to their resistance to stress. So if they're hard-bodied, they may not get crushed. If they have a high occupancy in the landscape, which means they can recolonize space, that's a resistance trait. And we can think about recovery traits, depending on how they move around, how mobile they are, what sort of larval phase they have. So we can classify the traits that these communities have into regulating traits. So that's the traits that give you regulating function, so restore functionality to systems and redundancy. And then we can think about response diversity traits, which are the set of traits that provide resilience to future disturbances or other stressors. Okay, so we can separate up those. So we can go through each of these species and characterize them, characterize the community in terms of the regulating traits and their response diversity traits. Okay, hopefully people are still with me. Then we can begin to think about how these networks are changed with stress. Okay, and trust me, this isn't me just with a line drawing things between dots. This is the results of a network analysis that Rebecca conducted on this data set. So we may have regulating trait A, so that might be, it's a deep burrowing species. Okay, that regenerates nutrients. We might have a response diversity trait A, which maybe it has a high occupancy in the landscape. And if the species shares those two same traits, we can draw a line between them, right? That's all we've done. So these little dots just represent a particular trait shared by a particular trait, either regulating trait or response diversity trait. So we can go through the entire network in a community and draw lines. The thickness of those lines represent the number of species that share that combination of traits. So the thicker the line, the more species that share that trait. And then we can do this, all the traits that we've got in our community, and draw something like this for rendergram. But the visual thing is to look at is that you can see there are some trait combinations that many species share. You can see other trait combinations that maybe only several species share. And so you can link all of this up. And again, it's the same idea. Networks that are complex, that have more connections, have more options for responding to stress and absorbing stress or recovery from other disturbances. Okay, so this is what we found. So when you do this, we found that the resilience of the system, so the resilience to maintain functionality, because that's one set of traits, and the response diversity, how it handles stress, another set of traits, was dependent on the background network complexity. So here's a control network that we built from a site that had a high network complexity site in the control. This is the one where that network complexity was more simplified. There was less connections, there was less functional traits in the community. Each of those little dots, remember, represents a trait, and there was less connections between them. And their response to the nutrient stress was dependent on that background complexity, either starting network that you began with. So in a system that was complex to begin with, and we added the medium nutrient treatment and the high nutrient treatment, you can see there's not a lot of change in that network complexity. It's absorbed that stress generated by the medium and the high nutrients, and it's sort of maintained it. However, if you take a control network complexity, where the network complexity initially was quite low, and then stress it through the nutrients, you can see that that has had an impact on the system and on the complexity of that network. It's more vulnerable to erosion of that resilience. So that background complexity was a function of the turbidity within the system, okay? In highly turbid systems, it tended to select for simpler network complexities, the initial starting point. For clearer sites, the control networks or the background networks look something like this. So again, we can begin to see how that turbidity stress is beginning to alter the complexity of these networks that makes it more vulnerable to an additional stress from nutrients. And this is kind of summed up in this plot here, and it kind of try and explain it in a way that hopefully people understand. So what we've got here on the x-axis is the control network complexity. So this is what in the control plots where we added no nutrients, this is what the complexity looked like. It's just an index saying if it's a larger number, there's more connections and thicker connections. As you go down that gradient, there's less connections and less species share them. And then what we've got plotted here is that change in complexity relative to the background complexity. So you normalize the change in complexity by what you started with to get some sort of magnitude relative percentage change. And this is for the high nutrients and the medium nutrients here. So the first take-home point from this graph is if you have high network complexity, then the medium and high nutrients don't really alter that complexity a great deal. It doesn't fall above that line or below that line. In the medium nutrient treatments, you can see as that network complexity gets simpler and simpler, the starting one to which we're adding nutrients, then the response becomes really variable. There's flickering going on. You can see there's a really positive effect of those nutrients at some sites where the network complexity increases, but at other sites, there's a really negative effect on that complexity. So the variance in the system goes up. And actually, if you're thinking about tipping point theory, one of the indicators of tipping point theory is prior to falling off the cliff, the variance in the system begins to increase. Then under the high treatments, you'll see as that network complexity increases, generally the outcomes are poorer and poorer. And then as it gets simpler, there's a much stronger negative effect on that network complexity. And so these systems, again, that turbidity stress, which is controlling that network complexity, that initial one, adding nutrients into it, affects the outcomes in these systems. So I think we've demonstrated just in those couple of studies that the role that that turbidity plays in terms of eroding the ecological network complexity and reducing the options and the capacity for stress. Remember these systems we're working in, if you were to go out and have a look at them, they wouldn't be highly degraded. We haven't worked across systems in our gradients that there's a mud pit over here and a beautiful sandy seagrass system over here. To look at these systems all look very, very similar. We've been working in a fairly narrow range of ecological health. They're all relatively good. We've just been looking at these subtle effects of these stresses. So the turbidity erodes that ecological network complexity, reducing the options for further stress. It alters the biodiversity ecosystem functioning relationships. And it also alters the resilient trait networks to future stresses as well. So as we're thinking about these kind of diffuse stressors across New Zealand and interacting them, we've also got to start thinking about a global context for this as well. And as we know, the world's warming and apart from heat waves and changing weather patterns, there's also going to be a profound effect on our coastal environment and particularly our estuarine environments, because they're right at the pointy end of climate change. We will build walls to protect coastal infrastructure, and building those walls means we're going to lose habitat or shift habitat in the marine systems. So a metre of sea level rise probably doesn't make a whole lot of difference for out in the open ocean where it's several thousand meters of water depth, but in an estuary where your mean water depth is only a couple of meters and most of your habitat is intertidal, it'll represent massive shifts in the proportion of area that's underwater versus areas of water that are above water for some of the time and underwater at other periods of time. And that really got me thinking about, given the importance of that hidden garden to our food webs, the productivity of these systems, how will the changing submergence times with sea level rise begin to impact on productivity, especially if that's coupled with poorer light climates associated with turbidity. So if you read the European literature, there's this idea in these systems that these intertidal habitats may provide some windows of opportunity. Okay, by windows of opportunity, I mean when the tide's in, the water's so dirty, there's no light hitting the seafloor, there's no primary production occurring. When the tide's out, there's sand flats and mud flats are exposed, and therefore you might have some resilience. There might be a window during those low tide sunny periods where the hidden garden, the microphyta benthos, can actually get some productivity underway, and there's some food to be passed on to the higher trophic levels. And so that got me thinking about the sort of changing sea levels and how that might alter the submergence emergence periods and how that may impact upon estuarine ecosystem productivity, the base of the food chain. So, habitat loss in New Zealand estuaries is going to be dependent on a whole lot of factors in terms of the existing morphology, in terms of whether people build seawalls, all of these kinds of things, retreat, etc. So this is a little data we pulled together from a number of the estuaries we worked in for that tipping points experiment, where we sort of put it through a metre or so of sea level rise to see how the proportion of that intertidal area would change. So some estuaries were quite resilient. You can see that we get 0.6 of a metre of sea level rise and then very little loss in intertidal area. Others, we began losing intertidal area immediately. Others, we sort of crossed a threshold and it would decrease very, very quickly. So the individual responses in estuaries are going to vary. But if these estuaries are turbid and they're dependent on that low tide production, then it's going to have quite severe consequences for the overall productivity of these ecosystems. So Georgina, for her PhD, began playing around with measuring photosynthesis irradiance relationships. So again, we've got our benthic chambers out on the sand flats. You can see this aerial image of them here. These sort of dark and light squares represent different shade cloth added onto the lids of those things, and we can alter the light climate hitting the sea floor. And then from that, we can derive whole ecosystem photosynthesis irradiance curves. So Georgina went out about 8 times over 2 years to get some seasonal information, and I've collapsed everything that she did over 2 years into one graph, and this is it. So this is the relationship between the photosynthetic radiation hitting the seafloor and the amount of carbon that's fixed by the system. And I've got 2 curves on here, one for the emerged production, so the low tide production that you measured when the tide was out, and one for the submerged production when the tide was in. So the classic photosynthesis radiance curve, the bands around them just represent the sort of seasonal variation that was measured. Interestingly, when we look at the breakpoints in that ecosystem function that we measured in the tipping points experiment across the whole of New Zealand, remember it's around 350 to 420 between clear and turbid estuaries. And that represents the band right here, that 350 to 420. And that just happens to fall right on the point where light really becomes limiting for microphyta benthos. So these breakpoints that we're seeing in functionality and their resilience to nutrient stress were associated, admittedly it's a correlation, but with the point at which light really begins to limit photosynthesis in the hidden garden. So what we can do with these kind of photosynthesis radiance curves is we can begin to play some thought experiments, what if experiments on how this might change if we alter the amount of submergence time, so how long they're underwater relative to how long they're out of the water, and also the relationship between the water column turbidity as it may darken it. The key thing to note for the microphyta benthos, of course, is that the emerged production is about a quarter of the submerged production. And that's because it's probably nutrient limited and desiccation is probably slowing it down. There's some, but there's not a lot. So we've been developing these little models where we look at based on the photosynthesis radiance curves, looking at kind of if we alter the amount of light in the water column. So this is at 100%, bringing it down to 0, highly turbid systems, and the proportion of time submerged. Here we are about 50% of the time submerged today, but that will decrease as the sea level begins to rise. So this little black dot here represents the current day conditions where we are today. So there's a couple of key take-home messages. If we keep the submergence time the same as in today, we'll see a decrease as the seafloor light climate goes down. You can see there's still some emerged production here, and that's sustaining the ecosystem at low tide production. If we increase the amount of time underwater and maintain good light levels, productivity will actually increase. Okay, we've got more time underwater, plenty of light hitting the sea floor. That's going to be good for business. However, if we have the combination of two stresses, where with climate change, increasing storm intensity, increasing inputs of sediments off the land, is coupled with a decrease in the amount of time that they're spending open to the air, then that decrease in that tail off of production will be very, very rapid. And these kind of relationships are really important because at the end of the day, you know, this microphyta benthos, the little hidden garden there what allows these godwits, which are just arriving from their summer breeding grounds in the northern hemisphere, to go from here to here in several months. It's the polyunsaturated fatty acids in the microphyta benthos that fatten up those birds and allow them to travel 13,000 kilometers in 10 days. Okay, and if we lose that intertidal habitat, we lose that production. There'll be a cascading consequences through the entire ecosystem, impacting ecosystems half a world away. So just a food for thought. All right, so just some quick take-home messages. I'm almost out of time. Our estuaries are highly valued and productive ecosystems. We're still lucky in Aotearoa, New Zealand. We're not in a state that many estuaries are in the northern hemisphere. There's still plenty we can do. The cumulative effects of sediments and nutrients are eroding resilience and making them prone to tipping points. However, local management action, so what we can do here and now, can help build resilience. If we can manage inputs from the land and we can keep the water clear, then that will provide resilience to climate change in terms of sea level rise, because that productivity at the base of the food chain will remain. And that's enough. Thank you.
We have time for questions, please. Oh, I have.
Conrad, so I guess my first question is, it's actually in the last sentence you said, which was managing sediment load. How do you do that?
Louis, no. I think, you know, it's a challenge, right? So we've been working with the MFE as part of the challenge and trying to, you know, it's a whole of society effort, right? You know, getting the right trees in the right place and you know, the right crops or the right animal use on the land, to riparian planting and things like that. And so, you know, we've been thinking a lot about what I'm kind of interested in exploring further is kind of, you know, everyone goes, oh, it's too expensive, it's too much, but there's lots happening, right? You know, Waikato Regional Council's been investing millions with local landowners to fence and plant, and all of things have to happen. But, you know, sometimes we get bogged down in the thought that, oh, you know, this is the most important place to do it, but we can't get it done there, and so we won't bother. But in actual fact, you know, there's probably, you know, as we talk about kind of the negative effects of cumulative effects of stresses, there's also the positive effects of small cumulative positive actions, right, that may build other things. So I think, you know, just, yeah, planting, you know, and better land use management really is the key. And we're still not going to get rid of all of it because climate and because just where we live. But you can have positive impacts on that. And if you reduce the sediment loading, there's a good chance you're probably going to help with the nutrient loading as well, because a lot of those actions will help with that.
Chris? Thanks, Conrad. There's some really awesome stuff in there. So based on your functional trait-based assessment of resilience, what are the chances of the right species with the right traits recolonizing those areas once you manage to see the nutrient inputs?
Again, I think it's pretty good. Again, it depends on what scale the degradation is at, right? So if you're just talking about kind of a local degradation in a small part of the estuary, then you've got other parts of the estuary that may be in better shape that can provide the recruits for that recolonization. If the entire estuary is trashed, and we do have New River estuary and Southland that's kind of reaching that point. It's basically been draining most of the Southland dairy intensification now for a number of years. That recovery is probably going to take a lot longer. And so, you know, the recovery process will happen as it happens, right? But I think, you know, most of the systems we've been dealing with, certainly the ones where this data has been taken from, you know, the management of the stresses should see positive responses. Remember, none of these systems have, you know, turned it into like some of the shallow lakes that you're dealing with around here, where there, you know, a lot of nutrients and a lot of hypoxia potentially. These are still healthy functioning ecosystems. These data are more around trying to get conversations with managers around the changes happening. You can't see it visually, but there is this erosion of the resilience through these systems. I don't know if I answered your question.
That was good. Good. Yeah, thanks.
Right.
The legacy effects of sediment, I guess if we did arrest sediment inputs from the land, it'd still be this, you know, ongoing problem.
Yeah.
Any thoughts about, you know, sort of the timelines?
Sea level rise might help us. Yeah, again, it's kind of, there's this triage thing, right? Like, and we've been talking about that a lot, about restoration, right? Because that's really where we have to go. And terrestrial and freshwater systems have been in that space for very long periods of time. But marine space, we don't have a how-to book with that. And the legacy, especially of sediments and estuaries, is a big one, right? So I think it's about kind of taking, with some systems you may do a little, and you know, it's a very long game, you know, intergenerational game for improvement, and then you may be dependent on a hurricane or something to flush everything out, or some, you know, some major change, but it still doesn't stop us acting now. But then you've got these other systems where are relatively pristine, where some careful management and some reduction in loading will provide that resilience and stop them tipping. So, you know, that's a question for managers in society, but you know, certainly the legacy effects of mud and of course all the feedbacks with mangroves and other things, you know, will be around for a long, long time, as the stored nutrients are in some of the lake systems as well.
So good. I got lots of questions. Compounding effects of additional stressors. You work with two.
Yeah.
You know, there's
Multiples
coming up. You picked the two obvious ones for New Zealand, but from a global context, when you put the other ones on top of it, acidification and temperature, how do you see those? I mean, do you see those as minor compared to nutrients and sedimentation, or do you see those as, you know, equally important in compounding? Because I mean,
yeah, and I think they're equally important in compounding, and sort of what I'm really interested in at the moment is the kind of non-lethal effects of climate change. Right. So you've got these systems that are already stressed by turbidity, so maybe their reproductive output from the animals isn't as high. They're hanging in there, so you haven't lost them from the system. They may be functionally dead, but then, you know, if you add a stressor on top of that, like a, you know, heat wave event, a lot of the estuaries, it's actually the terrestrial heat wave events that are more important than the marine heat wave events, because a marine heat wave event's only a couple of degrees. But what are the sublethal effects on the animal communities in these systems, where, you know, you may have reproductive failure because they're exposed to, you know, above average temperatures for a few weeks, and then there's no animals, babies coming back in. So I actually think trying to understand, and again, we're never going to understand it all. I see my role as kind of shining a light on at least some pathways that will get people to act and sort of change, because there's a whole lot of stuff we don't know, but that shouldn't stop us kind of trying to do the right thing now. And I think you're exactly right. I think when you put on climate change and increasing temperatures and acidification on top of nutrients and sediments, then we are going to be potentially eroding that resilience.
You look at that very simple network that you showed, it would be really easy to break one of those lines.
Yeah, it is.
Catastrophic.
Yeah.
I just want to point out that we also take questions over Zoom. So if people have questions, please type it in the chat.
Great talk. Really enjoyed that. Just wondering, I was really struck by that difference in photosynthesis and the submerged and emerged ones. And I was wondering, do those systems get really hot when they're exposed to the sun?
Yeah, they do.
And potentially limit?
Yeah, it does. I think, I've kind of been a bit not naughty is the wrong word. We're dealing with sand flats here. So if you actually go to Europe and you deal with the mud flats that they have, they characterize a lot of the mud flats, are the ones that you kind of sink to your knees in, right? Their intertidal production is way higher than what we measure, and that's because you have these microbial biofilms. They trap a lot of moisture on the sediment surface. There's tons of nutrients right at the sediment surface. So with our sand flats, because they don't have a lot of mud content, I think the water drains down, and so the plants are actually a little bit not light limited or temperature limited. They just can't get the nutrients they need to photosynthesize. And so I think that's why we see quite a drop off. So as part of Georgina's PhD, she also worked on seagrass systems as well, right beside the things. And the seagrass, man, they're going about 80 or 90% of what they can do underwater, out of water. So I do think it's probably in the situation that we're dealing with most of our estuaries here, where there's still sandy, not a whole lot of mud in them, then it's probably a desiccation and a nutrition as opposed to too much light and too much temperature.
So I have a question. So based on everything you've discussed, would you feel comfortable calling the phototrophs the keystone species for the entire?
I think they're the forgotten component in these estuarine ecosystems. So there's a great paper done by some people we know in the Netherlands a number of years ago now, where they demonstrated the intertidal production that occurs in the Wadden Sea, so a big swath of area off northern Germany and the Netherlands, actually fueled about 60% of the coastal biomass, subtidally as well as intertidally. Right, because that's where all the primary production is occurring. And so when Georgina started doing her PhD and Steph Mangan as part of her work, we went and did a literature review of photosynthesis irradiance curves, right? So you think, oh, light's important, people would study this, right? Given the importance of these microbes to photoautotrophs. We found 14 studies that had actually determined photosynthesis irradiance relationships for microphyta benthos, in the field or at least with intact communities that weren't kind of algal cultures or something like that. You do the same search for terrestrial systems, you get like several 100,000 hits, right? And so, yeah, they're kind of, their role in ecosystem, everyone knows they're important, but quantifying that importance and demonstrating the relationships with light and those kinds of things, I think are really important. It's funny, we had some comments back from Georgina's paper, and it was kind of like, well, it's not like that where we are. And, but their baseline has shifted to a really degraded state. And so where they only get low tide production. And, the point was actually, the productivity of these systems is way higher because of this submerged production is still supported, which is why we have so much shellfish.
A follow-up question. So I'm really curious because from what I can understand, the focus of these photoautotrophs for the field seems to be on your periodic ones. But the thing is, periodic photoautotropic organisms can actually use much less light than the periodic ones. So do you think, is there any understanding of the relative contribution of the periodic and periodic?
No, in a nutshell. Again, the fact there's only 14 studies of PI curves tells you a lot about how far down the line people are, in even identifying what's there and the relative roles of the. But you're exactly right. But I don't think, even, you're right that, if we do darken the waters, then basically you will get some photosynthesis occurring, but it won't nearly be at the levels that you've got with these guys.
Great. I think that's all.
Okay.
Conrad?
Peace out. Thank you. Do you want me to just stop?